Okay, so good afternoon to everyone. Thanks for joining today. First of all, I would like to thank the organizers for inviting me to be part of this fantastic series of talks about astrobiology. It's a real pleasure to be here today with such a great audience. So today in my seminar, I will talk about the input that the chemistry that takes place in space in the interstellar medium can provide to prebiotic chemistry and consequently to the possible appearance of life, not only in our planet, but also in other places in the universe. So the question of the origin of life has intrigued human beings for centuries, and it is still one of the great mysteries in science. The problem is intrinsically interdisciplinary and requires expertise in many fields, including biology, chemistry, geology, physics, and astronomy. This multi-focus approach, which is needed to add different pieces to the puzzle, is the main goal of my host institution, the Center of Astrobiology of Madrid, where researchers from very different fields are trying to understand better how life could be originated on Earth and whether it's possible somewhere else in the universe. So this is a simplified timeline of events pertaining to the early history of life on Earth. When we try to understand how life appeared on our planet, there are two general kinds of approaches. On one side, top-down approaches, which included phylogenetic trees or fossil records that tell us that life should have begun at least around 4 gigayears ago, more or less. This kind of studies face some limitations because the farther back in time we go, the less clear the evidence becomes. So at the end, we face a sort of informational horizon. As a result, we lack direct evidence for conditions on early Earth, including proof of the molecules that might have formed the building blocks of life. So complementarily, alternative bottom-up approaches have also been explored, which try to understand what could happen from the other side of this informational horizon. Since the formation of the Earth around 4.5 gigayears ago, like prebiotic chemistry, or even before than that, like astrochemistry. So although there is an intense and sometimes controversial debate about the origin of life, I think that we all agree that there are at least two very basic ingredients needed for life: water and organic material. Regarding water, the studies of a mineral called zircon found in Western Australia changed our initial view of early Earth as a magmatic shell and indicated that the planet had already a continental crust and oceans around 4.4 gigayears ago. So it seems that the presence of liquid water is not really a problem. But what about organic material? As indicated in this nice diagram, the origin of life required indeed a process of increasing molecular complexity from relatively simple molecules to larger biomolecules that already play key biological roles. So the question is, what simple chemical compounds could be realistically available at the dawn of life, and what was its origin? So one possible option explored by many authors is the endogenous synthesis, in which the organic compounds might be produced through atmospheric reactions on early Earth. An alternative approach could be the exogenous delivery, namely that these simple organic precursors arrived to the primitive Earth transported, well, at least some of them arrived to Earth transported by extraterrestrial objects. And this is certainly a plausible option, since we know that around 3.9 gigayears ago, our planet suffered a heavy bombardment of meteoritic and cometary material. And this timeline was indeed very timely, since this happens just before life appeared. So it is reasonable to think that the input of extraterrestrial material in the presence of oceans could play an important role for the development of the prebiotic chemistry that eventually ended with the dawn of life. In this scenario, is then very relevant to understand what kind of molecules could have been arrived from outer space. Going back in time, comets were formed in the outer part of the protoplanetary disk that originated our solar system. And this protoplanetary disk was born within a larger molecular structure, a larger molecular cloud in the interstellar medium. So at the end, this chemical journey led us to the study of the chemical composition of the interstellar medium, which is the field of study of astrochemistry. So we can define astrochemistry as the branch of astronomy devoted to the study of the census, the abundances and reactions of molecules in the universe and their interaction with radiation. One of the most exciting questions that astrochemistry is currently addressing is to determine whether the simplest molecular precursors of life are present in the interstellar medium, in the sites where stars and new planetary systems are forming. If this is true, the implications will be deep, since this will imply that the universality of the basic inputs for life, which means that life might not be a unique property of our solar system, but also of other sites in the universe. For instance, in some of the almost 5,000 exoplanets discovered so far in our galaxy. So during this talk, I will overview what we know about the chemical complexity that is possible in the interstellar medium. In summary, I will describe how we detect molecules in space, where in the universe we search for, what molecules we have found so far, and how we think that these molecules are formed. If one thing about it, the interstellar medium is a very hostile environment to allow chemical reactions, because it's really cold, its density is really low, many orders of magnitude lower than the air on Earth, for instance, and there are high levels of radiation, including UV, X-rays, and cosmic rays, and also violent mechanical impact due to the presence of shocks, for instance. So despite all these problems, which are not clearly helping chemistry to occur, the astrochemical studies in the last decades, and especially in the last few years, have shown that the interstellar medium is full of molecules and that the chemical complexity revealed is relatively rich. So since the detection of the first molecule in space in the late 30s of last century, the number of molecules detected, which is shown in this plot of the left, has continuously increasing. And as you can see, the increase has even accelerated in the last decade. Among the most of 250 molecules detected so far in space, more than 40 have been discovered only in the last 3 years. So we are currently witnessing a revolution in the field of astrochemistry, which have been possible thanks to the improvement of the sensitivity of the telescopes we use to detect molecules. So this is the list of molecules between 2 and 7 atoms we have been able to detect in the interstellar medium, but we have detected also many of the molecules that are more complex than that, with 8 or more atoms, many of which have been indeed discovered recently. And this list is increasing at an incredible rate. Not on a daily basis, but last year we have a huge number of molecules detected. So how we detect molecules? Before going into the details about some particular molecules that we have detected with a special prebiotic interest, I would like first to explain briefly how are we able to identify them in the interstellar medium. So the molecules in the gas in space exhibit different kind of motions, including bending, stretching, and rotations. And these motions produce emission of photons that we can measure with our telescopes. Each molecule has a different energy states, and when it goes from one state to another state of our energy, it emits a photon with a well-defined frequency corresponding to the energy difference between the levels involved. These photons can be detected thanks to radio telescopes working in the centimeter, millimeter, and submillimeter wavelength range. Since each molecule has multiple states of energy, multiple transitions are allowed following the rules of quantum physics. producing a characteristic pattern of emission at different frequencies. This pattern, you can see an example here that we call the spectrum of the molecule is unique of each molecular species. And therefore, the spectrum is like the fingerprint of the molecule, which allow us to identify it. Obviously, there is a crucial information that we need to know before trying to identify a molecule in space, which is indeed the rotational spectrum. This information can be measured in the laboratory by our colleagues, spectroscopists, which provide us the spectrum of the different molecules that we can then compare with the spectrum observed pointing our radio telescopes towards a particular position in the sky to identify which molecules are present there. So the basic principle is quite easy. In practice, the identification of molecule is a bit more challenging. I show you here a spectrum obtained by a spectral survey carried by the ALMA Observatory towards the star-forming region G3141. As you can see, we have a broad frequency coverage because we want to have as much molecular lines as possible to be sure that the molecule is really present there. But as you can see, the spectrum is completely full of lines, which is fine if we want to detect new species. But on the other side is a problem because this full spectrum is not only produced by a single molecule, but many. And the real challenge is to identify which molecules are there and disentangle their respective contribution. So this in black is the observed spectrum of this star-forming region in the sky, and this in red is the best model we have obtained after identifying tens of different molecules in this star-forming region. So at the end, what we can obtain is a full census of the molecules present in the region, but also we can derive their relative molecular abundances, namely how abundant are the molecules compared with others. And moreover, we can also obtain useful physical information about the region, like gas densities and gas temperatures. That's why molecules are commonly used in astrophysics as a powerful tools to study the physics of the systems, beyond pure astrochemical purposes. Okay, so despite the hostile environment, we know that molecules are formed in space, we can see them, but how are they formed? Without entering into details, there are two main ways to synthesize molecules in the interstellar medium. In the gas phase, through different mechanisms like ionization, dissociation, or two-body chemical reactions between a neutral and an ion, for instance, as you can see in the upper side of the slide. Or complementarily, molecules can also be formed on the surface of interstellar dust grains, which present some advantages that helps the chemistry to occur. To understand how a particular species is formed, what we usually do is to consider chemical routes that are possible in the extreme conditions of the interstellar medium. We introduce these chemical reactions into theoretical chemical models. I show you here an example of different chemical pathways that we studied some years ago for the formation of glycolaldehyde, a molecule which appears in many studies of prebiotic chemistry. We compare the molecular abundances predicted by the models with those measured in the observations, which helped us to better constrain what are the dominant routes, which eventually tell us how the molecule is formed. So as you have seen, astrochemistry itself is a field intrinsically interdisciplinary that involves the action of observational astronomers, theoreticians developing chemical models, and also experimental works in the lab, not only to provide the rotational spectroscopy of the molecules I mentioned before, but also to mimic in the laboratory the conditions we have in space, which is of great help when we try to interpret the observation. So where we search for when we try to discover new molecules? In our group, we are mainly interested in the different phases of star and planet formation, which are very nicely summarized in this slide. From the initial parental molecular cloud to the star-forming region where new planets will appear. And once we know where to look at, we need also to choose what molecules are our most interesting targets. So obviously, in the context of the chemical evolution related with the origin of life, we have focused our attention to the molecular precursors that could have been involved in the dawn of life. If we look into the narrow time period indicated here with a yellow box, in which we think that chemistry evolved to allow the jump to biology, the current general thought is that the last common ancestor, Luca, was preceded by a primordial DNA protein world. But there is growing evidence that points towards previous steps in which RNA played the dominant role, which is known as the RNA-world. In this hypothesis, initially proposed by the biochemist and physicist Walter Gilbert, RNA molecules could have performed at the same time the functions that nowadays are carried out by DNA and proteins. This RNA world could have existed as an intermediate step between prebiotic chemistry and DNA-based life. Very interestingly, a series of laboratory experiments in the context of the RNA world carried out in different research groups have shown that RNA nucleotides can be formed from quite simple molecular precursors. The figure here shows how the experiment by Becker 2019, was able to synthesize under possible earlier conditions both pyrimidines and purines from simple spaces located in the left side. So this is another pivotal work in the field in which Palmer and collaborators proposed a successfully chemical route to form RNA nucleotides, starting also from very simple molecules with a handful of atoms. Obviously, the full picture can be much more complicated than that, as nicely represented in this diagram here of the review by Kitadai and Murayama, but the message behind is the same. From simple molecular precursors, we can develop the chemistry needed for the RNA world. Therefore, these simple precursors are the molecules that we want to search for in the interstellar medium. We want to know whether all of these chemical building blocks are present or not in the interstellar medium, in molecular clouds and scaffolding regions, and consequently, if the interstellar chemistry can fit prebiotic chemistry. This is the main goal of our project, COOL, Cosmic Origins of Life. So let's start the search with a molecular cloud, in particular with the molecular cloud G plus 0.693 minus 0.027, which I will call for simplicity G0693. This cloud is located in the Sagittarius B2 region of the center of our galaxy. And this region is quite well known because it harbors an active protostellar cluster of massive stars that you can see below on the left, that are clearly visible in this image obtained with the ALMA observatory. Our molecular cloud is located at 1 arcminute towards north, where there is no sign of star formation activity. We think that this cloud will form stars in the future, but nowadays is in a pre-stellar stage. So molecular maps of the cloud have shown that the region is very rich in SiO emission, and SiO is a molecule that we astrochemists use as a very good tracer of shocks. If you have SiO, it's because you have powerful shocks in the region. So this emission is due to the presence of large-scale shocks in the cloud, likely from the interaction or the encounter of two molecular clouds. So a cloud-cloud collision that is producing these large-scale shocks. And this has important implications for chemistry, because the sputtering of the dust grains produced by these shocks are able to release into the gas phase the molecular content formed on the grains, enriching the gas phase and producing a very rich chemistry. That's why we are so interested in this cloud, because it offers us a great opportunity to the discovery of new molecular species. For this reason, we have been conducted in the last years an unbiased and very sensitive spectral survey of this molecular cloud using different radio telescopes. And so you hear some example of the spectra we got. So far, we have identified more than 120 molecules, which make this region one of the most chemically rich in the interstellar medium. To decide which new molecules we should search for, we went to the RNA-world precursors proposed by prebiotic experiments, and several of them were already detected in the interstellar medium, like formaldehyde, HCN, cyanoacetylene, cyanamide, or glycolaldehyde. There was a couple of key precursors that was elusive: hydroxylamine and urea. These two chemical compounds commonly appear not only in this particular experiment, but also in many other prebiotic chemistry works. So we decided to search for them towards the G0613 molecular cloud. And using our sensitive data of our survey, we were able to identify for the first time in space hydroxylamine, thanks to the detection of several of its molecular lines, as you can see here on the right panel. Encouraged by this detection of hydroxylamine, we also searched for urea, and we detected it as well. Although in this case, we arrived second because some months before, our colleague Arnaud Veloche published the first detection in the interstellar medium of urea towards the massive ethanolamine region Sagittarius B2N. So the important thing here is that two key precursors of RNA nucleotides, hydroxylamine and urea, are present along with others previously detected in the molecular clouds and star-forming regions, and at relatively high molecular abundances. In the RNA world, there is another agent that for sure played a crucial role: compartmentalization, which is a fundamental prerequisite in the process of the mergence of life, because membranes encapsulate and protect the genetic material and enable the metabolic activities within the cell. So the membranes of all current cells are made of a lipid bilayer of phospholipids, which are composed of a polar hydrophilic head and two nonpolar hydrophobic tails. I show you here on the right phosphatidyl ethanolamine, which is the second most abundant phospholipid in living organisms. If we take a closer look to the head, we see that the nitrogen-bearing head group is ethanolamine, a molecule that we thought that was potentially detectable in the interstellar medium. Indeed, the molecule was already being identified in the Alma-Ata Sita meteorite, which fell in the desert of Sudan some years ago. However, the region of ethanolamine was not clear, and two options were proposed: in situ thermal decomposition of amino acids under quite specific unusual conditions of the parent asteroid, or complementarily formation from smaller interstellar precursors prior to the formation of the asteroid. So then, encouraged also by the detection in a meteorite, we decided to search for ethanolamine in G0613 molecular cloud. The squeezed noise of the spectra allowed us to clearly identify at least 14 transitions of this molecule that are not blended with other species, which are shown in this plot here. Okay, so we know that the molecule is present there. We can also derive its abundance, but how it is formed in the interstellar medium. So using previous theoretical and experimental works from the literature, we have explored different possible chemical routes. We want to form this molecule here on the right, ethanolamine. A possibility is this hydrogenation sequence on the surface of dust grains proposed by Steve Charley back in 2001. Unfortunately, there is no current rotational spectroscopy measured in the lab of these intermediates, so we cannot search for them in the cloud. And unfortunately, these don't allow us to constrain better if this chemical route is possible or not. But these intermediates can be also formed from other known species already detected in the cloud. For instance, HNCCO can be from HCCO, or from chitin, H2CCO, or from HNC, all of them detected in the cloud. So they are possible and abundant, some of them precursors. As well, other of these intermediates might be directly formed through the different reactions proposed that I indicate here. In particular, the radical NH2, CH2, could play a key role, forming one of the intermediates of the hydrogenation chain combined with CO, or forming directly ethanolamine through combination with the hydroxymethyl radical, CH2OH. In any case, some of these different reactions are poorly known and deserve detailed studies, both in the laboratory and by using dedicated quantum chemical calculation. So the detection of ethanolamine in the interstellar medium has interesting implications for prebiotic chemistry. It is clear that to understand the origin of life, one should also understand first the origin and early evolution of cell membranes. It is thought that the very first cells were composed by relatively simple fatty acids and alcohols, as you can see here. The availability of ethanolamine in the interstellar medium and the detection in meteoritic material suggests that this molecule could have been delivered to the early Earth through meteoritic impacts. And this could have enabled the progressive replacement of fatty acids and alcohols by more efficient and robust molecules like phospholipids, playing a key role in the early evolution of cell membranes. Indeed, laboratory prebiotic experiments propose that phospholipids are formed using ethanolamine as a precursor under plausible earliest conditions. Moreover, given its similar chemical structure, ethanolamine has also been proposed as a direct precursor of the simplest amino acid, glycine, in simulated hydrothermal vents, considered one of the likely environments for the origin of life. But hydroxylamine and ethanolamine are not the only new interstellar molecules that we have detected towards the G0693 molecular cloud. In the last 3 years, we have detected up to 9 new species, which I show here in the slide. Between them, the Z isomer of cyanomethanamine, which is a HCN dimer that have been proposed as precursor of the nucleobase adenine, other imines like propergilamine, or more recently also 2 new amines, vinyl and ethylamine. Other colleagues are also working hard on the detection of new species and obtaining amazing results. In particular, 2 parallel spectral surveys of the TMC1 molecular cloud are showing that extremely rich carbon-rich chemistry, carbon chain chemistry, with the detection of rings like benzonitrile, you can see here on the left, or even double rings, like for instance indene, among other carbon chains summarized in this nice slide. So far, I have shown you molecules detected mainly in molecular clouds where there is no star formation activity at the moment. But what happened in regions in which we already know that stars are being formed? What happened in star-forming regions? These regions show also a rich chemistry, and we are then able to search for prebiotic precursors also there. In particular, I show you an example. We draw our attention to a species that was a key precursor of both lipids and ribonucleotides, according to the work by Patel 2015, and this is glyconitrile. So we point our telescope, in this case the ALMA observatory, towards the region IRIS 16293, which is a prototypical protostellar system with a rich chemistry. This image on the left shows you the dust emission which is surrounding the several protostars that are forming in this particular region. So previously in 2017, we detected an isomer of glyconitrile, methyl isocyanate, and this encouraged us to search also for glyconitrile. And we detected it successfully for the first time in the interstellar medium towards one of the protostars of this system. And this means that the gas that is surrounding the protostar is rich in a key RNA precursor. Therefore, we have seen that both in the parental molecular cloud and in the star-forming regions within them, molecules that are key precursors of the RNA world are present. This suggests that the chemical content can be inherited from one stage to the next one during the star formation process. I'm going to skip the protoplanetary phase because it would deserve another talk by itself, and I will address directly the chemical complexity in planetary system objects. One of the best ways to study the basic chemical ingredients that were present at the moment of the formation of our solar system is the study of the chemical composition of comets. In this sense, the European Space Agency mission Rosetta, which visited the comet 67P Churyumov-Gerasimenko, have completely revolutionized our knowledge about the chemical content of comets. The spacecraft Rosetta was launched in 2004, and 10 years later arrived to the comet and started to study it in detail from the orbit, and also with the lander Philae, which has a quite traumatic landing, if you remember that, but provided anyway useful information. The unprecedented in situ mass spectroscopy analysis of the mission have provided us a very rich catalog of molecular species, many of them detected in a comet for the first time, which was called the Cometary Zoo to honor its huge variety. So I show you here just a few examples of molecules detected, included thiols, alcohols, carbon-rich molecules, including rings, and one of the most interesting results from the astrobiological point of view, the detection of the simplest amino acid, glycine. But of course, although 67P is the only comet analyzed in detail in situ, there is not the only comet whose chemical content has been studied. Also, ground-based radio telescope observations have detected organic molecules in other comets, like in the case of the 46P with Tannen comet I show you here. So thanks to the wonderful instruments like ALMA and the Rosetta mission, we have gathered a rather complete view of the chemical content of both star-forming regions and comets, which indeed we can compare. How similar are the molecular abundances detected in the site of star formation compared with the comet 67P, for instance? And the answer is that they are reasonably similar. As you can see here in this plot, in which the molecular abundance of a low-mass protostar on the left, ID16-293 again, and in a high-mass stellar cluster, G3141, on the right, are compared with those of the 67 comet below. And as you can see, they are relatively similar. So this similarity of the chemical content in different interstellar sources have been also seen when comparing the relative molecular ratio of some organic species. I show you here the ratio between methyl formate and dimethyl ether, two molecules commonly detected in the interstellar medium in different kinds of sources. As you can see, the abundance ratio is close to 1 and very similar, regardless the type of source, from high-mass protostars, low-mass protostars, protostellar shocks, prestellar cores, galactic center molecular cloud, and also comets. This similar chemical contact during the different phases of star formation seems to confirm that the chemical reservoir is inherited during the whole process of the star and planet formation. Although, of course, this is a general statement, but many details are still to be addressed. So, and now in the last part of the talk, I would like to spend some words about the chemistry of phosphorus in the interstellar medium, in which we have been actively working in the last years as well. I think that within this audience, I think that there is no need to stress that phosphorus is a key chemical element for life, as was nicely summarized by the Chemistry Nobel Prize Sir Alexander Todd when he said, "When there is life, there is phosphorus." So phosphorus-bearing compounds, in particular phosphates, are so important for life because they have good chemical reactivity and at the same time they offer structural stability, which makes them perfect to build up macromolecules like ATP, phospholipids, RNA, and DNA. Indeed, the relative abundance of phosphorus with respect to hydrogen in living organisms is relatively high, for instance, around 10 to the minus 3 in bacteria. However, if we put on our astrochemical glasses and we go to the universe, the abundance of phosphorus is 4 orders of magnitude lower. The P/H ratio is around 10 to the minus 7. This means that the availability of phosphorus for interstellar chemistry is very limited. And this is why historically it has been really difficult to study molecules with phosphorus in space. Only a few detections of a single molecule, phosphorus nitride, PN, were published before 2006 in star-forming regions. That's why we decided some years ago, when I was still at the beautiful Centro Observatory in Florence, to conduct a large observational campaign with the 30-meter telescope, trying to extend the number of PN detection in massive star-forming regions. And we were able to detect PN in 22 of them. So thanks to these new detections in a large sample, we already obtained a very useful information. Phosphorus, we found that phosphorus is less locked on dust grains, frozen on the surface of the grains, than previously thought. And this means that we have more phosphorus available in the gas phase, which implies that we might be able to detect other species containing phosphorus using our observation. So with this in mind, we wondered if also phosphorus oxide was also present in the interstellar medium of star-forming regions. The interest of this small molecule is clear, since the chemical bond between a phosphorus and an oxygen atom is the very basic building block of phosphates, which are present in the structure of many important biomolecules, such, for instance, the double helix of DNA, as you can see here. So some previous observations tried to detect PO in star-forming regions before, but without success. So what we did was to select a couple of regions from our PNLR sample, those in which the emission of PN was stronger, expected that also PO was there. One of the sources is the W51 massive star-forming region that you can see here in this image. And we were lucky, and PO was identified for the first time in these two star-forming regions. And we found a quite surprising result. Because although PO is more difficult to detect than PN, its molecular abundance is higher than that of PN, and this was not really expected. This higher abundance of PO compared with PN has also been confirmed later by other works from our group and also from other colleagues. Since now I have shown you detection of phosphorus-bearing molecules obtained with single-disc telescope, which are telescope with a single antenna, which means that we don't have spatial information of the emission. And this information can be really useful when we try to understand better the chemistry and how these molecules are formed. So that's why we decided to use ALMA, which is an interferometer composed by many radio telescopes that also give us information about the morphology, the spatial information of the emission. And we decided to observe a stellar cluster called AFGL 5142. Here on the left, you can see the emission of another molecule, which is brighter, SO, which seems quite complicated maybe for a non-expert eye, but what we are seeing is quite simple. And it's shown in this sketch here. We have a massive protostar in the center that is opening a cavity in its natal core due to the presence of a bipolar molecular outflow indicated with the arrows. So this is again on the left emission of SO in three different colors that just indicate the velocity of the gas, but this is irrelevant here. What I want to show you here on the right is the map of the same region, but corresponding to the emission of PN. And here it is. This is the first map of a phosphorus-bearing molecule in a star-forming region ever observed. And as you can see, the PN emission is tracing very particular regions, the cavity walls of the molecular outflow, namely the interface between the outflow and the parental core. And this is giving us important information for understanding how these molecules can be formed. We have studied the chemical content of the different spots in which we have found PN and also PO is detected there. And again, PO is more abundant than PN in a factor between 2 and 8. And very similar values has also been found recently in other maps towards a low mass protostar by Werner et al. Early this year. So it seems clear that PO is more abundant than PN in star-forming regions in our galaxy. Okay, so we have learned more about the phosphorus-bearing molecules in the interstellar medium, and the next natural step was to learn about its possible connection with that of the solar system. And to do that, we used the amazing data of the comet 60P Churyumov-Gerasimenko from the Rosetta mission. In a paper published by Catherine Alweth back in 2016, the mass spectroscopy of the instrument Rosina clearly identified a peak corresponding to atomic phosphorus, but at that time was not clear if this phosphorus corresponds to molecules that we are seeing in star-forming regions like PO or PN, or to other more complex phosphorus-bearing molecules. So Catherine went back to the data, and she identified a clear peak mass at the position of PO. So most of the PO, we know now that most of the PO of the comet is in the form, most of the phosphorus in the comet is in the form of PO. So we also searched for other species like PN, PS3, CP, and even more complex phosphorus-bearing molecules, but none of them is detected. So we derive upper limits for their abundances. So in the comet 67P, the ratio between PO and PN is higher than 10. So again, PO is more abundant than PN, as occurs in star-forming regions. We think that PO, like other volatiles detected by Rosetta, was already in the cometary ices prior to the birth of the sun, and hence that the chemical budget of the comet was inherited from the natal environment of the solar system. So the similarities of the chemical content of star-forming regions and the comet 67P allow us to speculate how the interstellar threat of phosphorus might be. There is no growing evidence pointing that our sun was born in a clustered environment in the company of other stars, including also massive stars. And this environment is indeed quite similar to the one we have observed in the IFGL 5142 stellar cluster, where PO is the main carrier of phosphorus in the gas phase, like occurs also in the comet, whose chemical composition could be inherited from the natal environment of the interstellar medium. As I have mentioned before, comets may have delivered significant amounts of prebiotic material to the early Earth, and in this context, the PO-containing comets could have contributed to the prebiotically available phosphorus reservoir during the dawn of our planet. This is a possibility, but of course, other possible sources of extraterrestrial phosphorus have also been proposed, like in the form of riverside contained in meteorites. And to conclude, what happened beyond our galaxy? We also have phosphorus-bearing molecules in other galaxies. And to answer this question, we have analyzed data of an unbiased and sensitive spectral survey obtained with a large program of ALMA called Alchemy, which is focused on the detailed study of the molecular inventory of the galaxy NGC 253 that you can see on the left side. The first results of the project have already provided a complete list of molecules. And regarding phosphorus, we have also detected for the first time outside our galaxy a molecule containing phosphorus. In this case, we detected PN. This expands also even more until other galaxies the horizon of the power of interstellar chemistry in producing molecules with prebiotic relevance. This stresses the idea that the basic chemical ingredients for life are distributed across the universe. So to conclude, I leave you with the takeaway message I would like that you bring home. The chemical complexity of the interstellar medium is rich, and it offers a possible feedstock for prebiotic chemistry, in particular, providing key chemical precursors of the RNA-world. And finally, I would like to advertise a session that we are organizing about the astrochemical heritage from molecular cloud to planetary surface that will be held during the next European Astronomical Society annual meeting in Valencia, Spain, at the end of this June. Please join us. It's going to be a great session. And thanks for your attention. I will be very pleased to answer questions.